Our Globe TVers breaking news. Yes, disease-causing bacteria of the genus Salmonella or Yersinia can use tiny injection to apparatuses to inject harmful proteins into host cell, much to the discomfort of the infected person. However, it is not only with a view of controlling disease that researchers are investigating the injection mechanism of these so-called type 3 secretion systems, also known as injectisomas. If the structure and function of the injectisomas were fully understood, researchers could hijack it to deliver specific drugs to cells, such as cancer cells. In fact, the structure of the injectisome has already been elucidated. However, it remained unclear how the bacteria load their syringe so that the right proteins are injected at the right time. In a study published in Nature Microbiology, a team of scientists led by Andreas Diepold from the Max Planck Institute for Terrestrial Microbiology in Marburg and Ulrich Endensfelder from the University of Bonn has now been able to answer this question. Yeah, mobile components of the injectisome come through the bacterial cell in search of the proteins to be injected. So-called effectors. When they encounter an effector, they transport it like a shuttle bus to the gate of the injection needle. How proteins of the sorting platform in the cytosol bind to effectors and deliver the cargo to the export gate of the membrane-bound injectisome is comparable to the processes at a freight terminal, explains Stephen Wimmy, first author of the study as a postdoctoral researcher in Andreas Diepold's laboratory. We think that this subtle mechanism helps to make the injection efficient and specific at the same time. After all, the bacteria have to inject the right proteins quickly to avoid being recognized and eliminated by the immune systems, for example. To gain this insight into important loading mechanism of the injectisome, the researchers had to apply new techniques, conventional methods, which are normally used to detect that proteins bind to each other, did not work to answer this question. Possibly because the effectors are only bound for a short time and then immediately injected, explained Andreas Diepold, research group leader at the Max Planck Institute and co-leader of the study. That's why we had a analyze this binding in situ in the living bacteria. To measure this transient interaction, we made use of two novel approaches that work in living cells: proximity labeling and single particle tracking. As Ulrich Endensfelder, whose group worked on the study in 3 different locations: the Max Planck Institute in Marburg, Carnegie Mellon University in Petersburg, and the University of Bonn. Beyond the report, Glory.